Good day, student, for the school subject biology, ACSC. This tutorial will be very imperative in guiding you to what? The preparation of your examination. So please, I'm really asking you to pay attention and also to just try to understand and read through carefully the guidelines that will be given to you today. Very good. Let's start. We are starting with our topic on diversity of living organisms. This topic of diversity of living organisms, we have, for example, the kingdoms, the five kingdoms. The five kingdoms where we are looking at kingdom bacteria, kingdom protoctista, kingdom fungi, kingdom animalia, and kingdom plants, where you have to look at the diagnostic features. The diagnostic features meaning that if you look at, for example, kingdom animalia, and you look at, for example, an animal, it can be a baboon, it can be a human being, it can be a giraffe. You look at the visible features. The diagnostic features, we are referring to the visible features that you see on that organisms. Only the one that you can see. For example, their body are covered with fur or hair. Also, you look at the pinnae or external ear flap, the mammal gland. Those are some of the features that are distinguishing kingdom animalia from the rest of the kingdom. So not only the kingdom animalia, you need to ensure that you understand the kingdom bacteria, their features such as they are unicellular. Unicellular meaning that these organisms, the bacteria, are only made up of one cell. They don't have a nucleus. Their cells lack nucleus. That's why they are called prokaryotic cells. Also, you have to look at in terms of they don't have mitochondria, and also they don't have any organelles that is surrounded by a membrane, meaning that they don't have mitochondria, as I said earlier, they don't have endoplasmic reticulum, they don't have a chloroplast. Any organelles that is surrounded by the membrane is not present in the kingdom bacteria. So those are some of the features that you have to look at. Looking at kingdom protoctista, the kingdom protoctista, these are some of the organisms that have a little bit advanced in terms of the features. They are a little bit complex in comparison to kingdom bacteria. They have a nucleus. They are visible, meaning that they are made up of many cells. They are multicellular, although we have some exemption. We go to kingdom fungi. You look at the fact that the kingdom fungi, this is where we have organisms. They have cells which is made up of cell wall, and their cell wall contain chitin. These organisms, their body are made up of hyphae, like thread-like structure called hyphae, and also they have a sporangium, and that sporangium contains spores. When it reaches certain size, it disperses, and those spores then form another fungi, meaning that they reproduce asexually by a means of forming spores. So looking at kingdom plants, again, you have to look at the features in terms of that they have leaves, they have organs like leaves, stems, roots, and you really have to distinguish that the kingdom plants, one of their features is that their leaves contain chlorophyll, which is found in the chloroplast. That's why they are autotrophic, meaning that's why they are able to produce their own food. So the diversity of living organisms is not only looking at the kingdom. You have to look at the kingdom, you have to look at the phylum, both chordata and invertebrates. For example, the arthropods. The arthropod you have to look at in terms of their features. Again, they have three main features, all arthropod, regardless whether that arthropod is an insect, whether it's a crustacean, whether it's an arachnid, or whether it's a myriapods. They both have segmented body, meaning that their body are divided into sections. Also, they have jointed legs. Also, they have an exoskeleton, a hard exoskeleton. Then we go back to individual classes, 4 classes: the insect, the arachnid, the crustacean, and the myriapods. You need to look at their features in terms of, for example, the legs. How many legs are found in all insect? How many do they have wings? For example, when you look at class insect, some of them have wings, some are wingless. So we look at again in terms of their body parts. For example, the insect, they are divided in 3 body parts: head, thorax, and abdomen. But when you look at arachnid and crustacean and myriapods, their body are divided into 2 sections, meaning that the head and the cephalothorax. The cephalothorax is a part that is made up of thorax and abdomen. So you really need also to look at the diagnostic features of the arthropod in terms of insect, arachnid, crustacean, and myriapod. In myriapod, we have sub-2 classes, which is centipedes and millipedes. Let's look at the next one, the light microscope. The light microscope, you need to look at the parts of the microscope in terms, for example, from the eyepiece, body tube, objective lens, the stage clip, and also the function. For example, the function of objective lens is to magnify the object that you are viewing. The object meaning the specimen that you are viewing. Whether you are looking at the plant cells, whether you are looking at an animal cells, or whether you are looking at a parasite, as long as that tiny object, it is magnified with the objective lens. Preparation of microscope slide. You need to know how to prepare it. You need to make sure you need to have a slide. You need to have a slide cover. You can, for example, look at the onion. You peel out the thinnest part of the onion. Then you put it gently on the slide at a 90 angle. Then what you do, you add a bit of water. You cover it with a microscope slide. And then you gently place it on the stage. And you use the microscope starting with a smaller, not smaller, but we say the lowest objective lens until you are able to view the image clearly. The type of cells. The type of cell here we are referring to, we have 2 types of cells, meaning that all living organisms, whether bacteria, whether protoctista, whether fungi, whether animalia or plants, they are grouped into 2 types of cells, either prokaryotic cells and eukaryotic cells. So prokaryotic cells, very, very significant. The moment you hear the word prokaryotic cells, these are cells that do not have a nucleus. The eukaryotic cells are cells that have a nucleus. Again, the prokaryotic cell, no bounded membrane, meaning that no bounded membrane, they don't have a structure that is surrounded by a membrane. And again, when you are comparing, if you say on a prokaryotic cells, no membrane bounded organelles, you must again look on the other side of the eukaryotic cells and say, it has a membrane bounded organelles. So your comparison between these 2 types of cells, if you give, for example, no nucleus on a prokaryotic cells, on the other side of eukaryotic cell, you must say it has a nucleus. So meaning that it must correspond, it must be opposite to each other, and that only worth 1 mark. Each difference worth 1 mark. Let's look at photosynthesis. Photosynthesis it's a process that is carried out by all organisms that contain chlorophyll. We need to pay attention that all organisms that contain chlorophyll. We cannot just say plant because we do have some protoctista such as euglena that contain chlorophyll. As long as that organisms have chlorophyll, it has an ability to trap light energy and convert it into chemical energy. In that process, where that specific organisms produces its own food, we call it photosynthesis. Eutrophic, what is eutrophic? It's a type of feeding. What do we mean? This eutrophic means these are the organisms that can produce or make their own food. Again, why are they able to make their own food? This is because they contain chlorophyll, which is located in the chloroplast. The equation for both in words and chemical, equation for photosynthesis, please, in words. For example, the equation for photosynthesis, you start with carbon dioxide plus water, and you have an arrow. On top of that arrow, you write light energy below chlorophyll. The arrow indicating the product, which is glucose plus oxygen. And then the chemical equation, you need to pay attention. Again, you just write it. Carbon dioxide, you write it in symbol CO2 plus water, you write it in symbol H2O. Then an arrow, and again you must write a light energy. Light energy cannot be written in symbol, but it must be there. So the same as chlorophyll. What is produced is C6H12O6 plus O2. Then you balance the equation, meaning that the number of elements that are present before the arrow and after the arrow, they must be the same. For example, when you look at the number of elements before the arrow, you have carbon, which is on the carbon dioxide. It's only one, but you have 2 oxygen. So now you need to make sure and on the other side, you have 6 carbon, which is on the glucose. Now you need to make sure which number can you look for, which will give you also 6 carbon. On the other side before the arrow, it's 6. 6, and you add 6 to carbon. You also add 6 to H for water, and you also H, which is at H2O, and you also add 6 at oxygen. At glucose, you don't add anything, and you have your balance equation. Very good. We look at external and internal part of the leaf. You look at any leaf in your environment, you need to understand which one is a leaf apex, leaf petiole, the leaf blade, and the middle line, which is on the leaf, which is called the leaf midrib, as well as their function. We look at the internal. Internal means the inside part of the leaf. We start with a cuticle or cuticle, some may pronounce it like that, and it is made up of fat. That's why it reduces the loss of water through transpiration. Below the cuticle, we have parasite cells. The parasite cell, now you need to know that there are 2 parasite cells, the one that are located on the upper side next to the cuticle, and the one that are located on the lower side. Now, after the upper epidermal cells, in the upper epidermal cells, their role meaning their function. They are there to ensure more light penetrate into the lower cells. The lower cells, for example, the parasite cell and the spongy mesophyll cell. Going back to the parts that make up the internal part of the leaf, below the cuticle, we have the parasite cells. The parasite cells they have a rectangular shape. In majority of biology textbook, they usually show a plant cell. That plant cell that in majority of the biology textbook, it's basically a parasite cells. There are many in the plant leaf, and they contain numerous or many chloroplasta. The reason why they contain many chloroplasts is because this is where photosynthesis takes place the most. From there, you need to look at the cell below, which is spongy mesophyll cells. Between the spongy mesophyll cells, you notice that there is a space. That is a space. That is basically to facilitate the diffusion of carbon dioxide into either the parasite cell or the spongy mesophyll cells, because carbon dioxide is needed as a raw material for photosynthesis. If we go back to the photosynthesis equation, we start with carbon dioxide, meaning that carbon dioxide must be there. We go further and we talk about the guard cells. Again, between the guard cells, we have the stomata. The stomata are controlled by the guard cell. What does that mean? Meaning that the function of the guard cells is to control the opening and the closing of the stomata. Let's move on. We go to the breathing system. We start with parts and functions. The part, you start from the nose, the nostrils. We go to the trachea or a windpipe. We go to the bronchus, meaning that the tube from the trachea or the windpipe, if it's one, it's called bronchus. If there are two, meaning that if your labeling indicate two lines, and they come at the end, it merge into one, then you can't say it's a bronchus. You must say you need to write it down as bronchy, because that indicates the plural. Then we look further at the bronchiole. From the bronchiole, at the end of the bronchiole, we have a round-like structure called alveolus. If it's one, it's called alveolus. If there are two, it's called an alveoli. Then we need to look at in terms of the function. When you look at the function of the trachea, the bronchus, the bronchiole, they are all air passage. What does that mean? Meaning that they all allow air to enter our breathing system or to leave our breathing system. The alveoli, the function of the alveoli, this is where gases exchange take place. What does that mean? This is where oxygen coming from the bronchiole diffuses out of the alveoli into the blood capillary. And at the same time, carbon dioxide from the blood capillary, which is coming from the tissue as a product of respiration, carbon dioxide is diffusing from the blood capillary containing blood into the alveoli, and they are diffusing out of the blood capillary because the blood capillary contains blood which is rich in carbon dioxide. So diffusion will occur there from a high concentration of carbon dioxide to a low concentration of carbon dioxide in the alveoli. At the same time, oxygen is diffusing out of the alveoli into the blood capillary because there is a high concentration of oxygen in the alveoli than in the blood capillary. Good. So you need to look at the alveoli also in terms of the structure and also in terms of what adaptation. How are the alveoli adapted to perform their function? The alveoli, for example, they are numerous, meaning that there are many. And this increases the surface area so that maximum gases exchange can take place. Also, they have a thinner cell membrane. Remember, not a cell wall, because animals do not have a cell wall. And when you look at the alveoli are found in animals. Good. So you also need to look at other features of the alveoli. For example, the moisture. For example, they are surrounded by the numerous transport system, which is the blood capillary. The next thing, the effect of exercising in the breathing system. What does exercise do to your breathing system? Does it increase the depth or does it make you breathe faster? So it actually increases both. It increase the depth and it make you breathe faster. This is so that you can supply more oxygen to your tissue and at the same time to get rid of more carbon dioxide, which is produced by respiration, because there is a faster respiration because your body is involved in an activity. Your body needs energy. And for that energy to be produced, it needs to be produced through respiration. So there's a faster respiration taking place in your body. So at the same time, the product of respiration is carbon dioxide. So more carbon dioxide is produced in your body, and that's why you need to breathe out faster to get rid of carbon dioxide, and again to breathe in faster to supply your body, your muscles with more oxygen. Good. We look at sexual reproduction in plants and animals. We look at sexual reproduction in plants in plants and animals. Take note, plants, they have an ability to reproduce either sexually or asexually. But in this case, we are focusing on sexually. So we are looking at mainly on the organ called the flower. You need to know the different part of the flower. We start with a petal, the sepal. Then we go to the male part. The male part is called the stamen, which is made up of the anther and the filament. And you also have the female part of the plant, which is called the carpel, which is made up of the stigma, the style, as well as the ovary. Inside the ovary, there is an ovule. You need to look at fertilization in plants. What does fertilization mean? It's just a fusion of the pollen grain, which contain the male gametes, and the ovule, which contain the female gametes. That fusion will result in a formation of a seed. And that, because you have two gametes that are involved, the male gametes and the female gametes, the fusion, it's the fertilization of these gametes. And the formation of these seeds through the fission of two gametes from two different species, male and female, it's called sexual reproduction. So you also need to look at in terms of the seed that is formed. Also, animal, we need to look at the structure, the female reproductive system, the ovary, the oviduct, the uterus, the cervix, the vagina. And you also need to look at in terms of what is the function of the ovary. It's responsible for producing egg cells, but it's also responsible for producing the sex hormone, estrogen, as well as progesterone. We look at further to pollination and the type of pollination. Take note, I spoke about the flower earlier. Pollination is a transferring of pollen grain from the anther to the stigma. The type of pollination, we have cross-pollination and self-pollination. We have to look at in terms of the advantage and also the disadvantage. The advantage, for example, of self-pollination is that it allows these new species that will be formed, the plant species, to be similar, to be identical. We go further to transpiration. Transpiration is a loss of water through tiny holes on the surface of the leaves called stomata. So the loss of water, when a plant loses water, the loss of water allows a plant to absorb water through its roots. Factor influencing the rate of transpiration, you have to look at in terms of temperature, the wind speed, the light intensity, and also you have to look at in terms of the size of that plant leaves. How wilting occurs in plants? Wilting is when a plant loses more water than how much water, than the amount of water the plant can absorb. And that's basically it. Xerophytes. We, especially in Namibia, we are blessed to have xerophytes that we can observe around. Xerophytes are basically plants that are adapted to the arid condition. Our camel thorns, our Welwitschia mirabilis, our quiver tree, all these plants are adapted to live in a very dry condition. Arid there means dry condition. The next thing that I would like to talk about is this topic is on the nervous system. This topic has to do with the synapse cells. This topic is about how the nerve impulses move, or we use the word transmitted, from one neuron to the other. You learn that there are three types of neuron: the sensory neuron, the relay neuron or the interneuron, and the motor neuron. Now, we are looking at the terminal. Terminal means at the end of either sensory neuron with a sensory neuron, or a sensory neuron with a relay neuron, or a relay neuron with a motor neuron. We need, what is a synapse? It's the end of the two neuron, the end of two neurons. Now, the neurons, they don't touch each other. There is a space between. As you can see there, the space called the synaptic gap. So that is one structure that make up the synapses. Another structure that you can see there, it is what is called the mitochondria. And this mitochondria is found in a presynaptic neuron. Remember pre-primary, like before you start primary. Now, the synaptic, looking at the synaptic gap, it's a gap between the two neurons. This neuron before the gap, we call it presynaptic neuron. So this is the end of the axon for the presynaptic neuron. It also has a presynaptic membrane. Moreover, the end of axon for the presynaptic, which is this one. Why are we calling it the end of axon for presynaptic neuron? Because it is before the synaptic gap. And the one after the synaptic gap, we call it the post-synaptic neuron. Post, like when you graduate from university, the second qualification, they call it a post. You are a postgraduate. You are not an undergraduate. Post means after. Post-synaptic, after the synaptic gap. Now let's look at, because we need to know the structure. Again, reflecting back, we say the synaptic is made up of synaptic gap. That is a gap between the two neurons. It also made up of the end of axon for the presynaptic neuron. It is made up of this swollen, we call it a synaptic knob. It is also made up of mitochondria. The membrane around the presynaptic neuron, it is called the presynaptic membrane. What is changed there is just the neuron, and you look at the membrane. What is also found, another structure that is also found, only in the presynaptic neuron, it is the it is the synaptic vesicle. The synaptic vesicle is a round structure which contains the dot-like structure inside, and the dot-like structure are called neurotransmitter molecules. The neurotransmitter molecules have names, and in your book there are names of those neurotransmitters. And this neurotransmitter, once the nerve impulses reaches the end of the presynaptic neuron, then this vesicle, it bursts. And once it bursts, it releases the neurotransmitter across the synaptic gap, meaning that the neurotransmitter diffuses across the synaptic gap. And these neurotransmitter, they carry the nerve impulses from the presynaptic neuron to the postsynaptic neuron. On the other hand, when you look carefully on the structure of the synapses, the postsynaptic neuron, it is the only one that have a receptor site. The receptor site is for what? To receive the neurotransmitter. And the presynaptic neuron is the only one that has the synaptic vesicles with a neurotransmitter. This ensures that the transmission of nerve impulses is only a one-way. Because one-way meaning that the nerve impulses are moving from the presynaptic neuron to the postsynaptic neuron. Why are these nerve impulses moving in one direction only? It's because of the structure that make up the presynaptic neuron. The structure of presynaptic neuron is the only one that have the neurotransmitter. But the postsynaptic neuron, it does not have the neurotransmitter. The neurotransmitters that are there are diffusing from the presynaptic neuron. Another thing is the receptor site. The receiving structure is only on one side, on the postsynaptic neuron. That's why it's the only one transmission. It's a one direction. It's not a two-way direction. It's a one-way direction. Good. We move on to the next slide. We look at the effect of opiate drugs on the transmission of nerve impulses across the synapses. The effect, what are opiate drugs? Opiate drugs are drugs that slow down the transmission of nerve impulses and make you numb. These drugs can be used, for example, in surgery, or when the doctor wants you to sleep, so that you don't feel the pain when they are carrying out a certain surgical procedures. So what they do, they slow down the transmission of nerve impulses. That is the effect that it has. We look at the structure of the retina. The retina is a structure that is found in our human eye. Not only human eye, all the mammalian eye, they do have a structure called the retina. When you look at it, we can look at it in terms of the structure, we can look at it in terms of, it forms a third layer. It forms a third layer of our eyes. If we have to look at the layer from outside into the eyes, it's the one that forms a third layer. And it consists of 3 regions. And also, it is further in front, attached to the serial and suspensory ligament. There are also neurons that are found in the retina. The neurons that are either 3 or 2 that hold a certain structure in the retina. So we need to understand carefully those structures well. We look at the menstruation cycle. The menstruation cycle, you should be able to read to know in terms of what is the effect of estrogen and progesterone on the menstruation cycle. After the menstruation, what is the function of estrogen? Estrogen is there to repair the uterus lining. It's there to make the uterus lining thicker. It's there to ensure ovulation take place. So, as well as the role of progesterone, it is there to make sure to maintain the uterus lining just in case if fertilization occurs. It also ensures that you don't menstruate, for example, immediately after you had menstruate. Contraceptives are commonly used in Namibia. So we have contraceptives that are used in Namibia. We have, for example, we have the IUD, which is the uterine device. It's also called a loop, which is inserted into a woman's uterus. We have condoms. We also have the different forms, the jelly or the spermicide that kills sperms. We also have what we call the diaphragm. The diaphragm, it's used, a woman is inserted into the private part and block, and it fit perfectly the cervix. The idea is to block the passage of semen. Also, you have to look at in terms of which one is more effective and what is the purpose, what is the risk. For example, the risk in terms of the sexual transmitted disease for the diaphragm, for the IUD, for the spermicide, for the different pills. The risk is that it's not 100. It doesn't protect a person against sexual transmitted disease. It doesn't protect a person from HIV and AIDS. Also, we have to look at in terms of, is it effective? Very effective when it comes to the condom. When it comes to the pill, it is 95% effective. And also, when you look at, for example, in terms of the IUD or the loop, very effective. But sometimes it might fall out without knowing. And another thing, when it comes to diaphragm, one need to know how to insert it in, because if you might be in a rush and you insert it in and it did not fit properly or it did not close the cervix properly, pregnancy may occur. We also have to look at STI. This is sexual transmitted infection. These are some of the infections that are transmitted through sexual contact. We have common one in Namibia, the gonorrhea, the syphilis, as well as the HIV/AIDS. In terms of, you need to look at the symptoms. In terms of the treatment, is a treatment for them? In terms of the curability, is it curable? How do we protect ourselves? Genetic terminology. Genetic terminology, we are referring in terms of all the terminology starting from genetic. What is genetic? It's a study of inheritance. It's a study of how information are passed on to their parents. We went further, and in terms of, you look at the terms of what is a gene. It's a section of a DNA. You went further and you look at what is an allele. An allele is an alternative form of genes. You went further and you look at homozygotes, heterozygotes, recessive, dominant, homolozygotes, genes. You have to look at in terms of all those definition, and you also have to look at what is codominance. For example, of a codominance, these are whereby when the genes, all the genes have an effect in an organism. For example, the blood group. The blood group is one of the example. An example is it's a mating. We might not really say it a mating, but it's a reproduction between a black individual, a person with a black color, and a person with a white color. So the child tend to be in the middle, not black, neither not white. So that means both of these individual, their genes are equally strong, and that is what we call codominance. Variation. When you look at the person next to you, for example, that individual look different from you, and sometimes that individual is your siblings. We have differences. Variation refer to the differences that occurs among species, individual differences that occur among individuals belonging to the same species. So you can't compare yourself to another species, but you need to compare yourself to the same species. The person next to you, you look at the variation. Now, we have two types of variation: continuous variation and discontinuous variation. Continuous variation, a type of variation that is merely caused by the environment. For example, the height, it's caused by the environment and the genes. Very, very important to notice that. So continuous variation is a type of variation that is a combination of, it's a difference that is caused by the genes that you have, that are in your cells, in your nucleus, plus the environment where you are. The height, the skin color, the behavior, those ones. On the other hand, discontinuous variation are the type of variation that is only caused by the genes. For example, the ability to roll your tongue. You can either roll your tongue or not. Your blood group, you either have blood group A, you don't have a blood group which is in the middle. So the discontinuous variation is where you have distinct category, where you are either A or B. You are either a male or female, not something in the middle. Now, ecosystem, that is the chapter that we also have to look at. Ecosystem, we look at in terms, we need to look at some of the terminology there. What is ecology, what is ecosystem, and how living and non-living component interact with each other. So we need to look at the producer, the consumer. Always when we're talking about a producer, we are referring to plants. Plants are producer, meaning that they produce food. Consumer are animal that consume or that eats food. That food can either be from plants or can either be from themselves, animal. Now, we have now different type of consumer. We have primary consumer, secondary consumer, tertiary consumer, and quaternary consumer. And the last one, we have a decomposer. This is also where we have a food chain and food web. Please take note. Whenever you are starting your food web or food chain, it must always start with a producer. Some people start with a son, it's still fine, son, but the next one must be a producer. And from a producer, the primary consumer. From a primary consumer, secondary consumer. From a secondary consumer, tertiary consumer. From a tertiary consumer, quaternary consumer. From a quaternary consumer, in that case, can be a decomposers. Very good. The type of competition in ecosystem. In an ecosystem, you have different animals. These different animals are eating the same food. They are looking, they have the same mating partner. So there is a competition in an environment, competition in terms of food, competition in places where they are habitant, and also competition in terms of their mating partner. So you need to look at, we have in our textbook, we have two types of competition that are indicated there. We need to know them. We also need to look at advantage and the disadvantage of organic fertilizer. Now, this component of agriculture is part of biology, and we need to look at the organic fertilizer, our fertilizer that are obtained. We obtain either from plants or animals. These are the fertilizers that are natural. They are made from plant or animal. They are not artificial. Now, advantage of organic fertilizer, they improve the soil structure. Of course, they improve the yield meaning that they make the plant to grow, but they play a very important role in the improving of soil structure. They also help with water infiltration, meaning that they help to allow water to move into the soil. Another advantage of organic fertilizer is that they help in the water holding capacity of the soil, meaning that if a soil has organic fertilizer, that soil hold on to water longer. So that means that soil will not dry faster, unlike in comparison to inorganic fertilizer. In that plant, in that soil where such plant is growing, it will grow because it keep water longer. We look at the disadvantage. The disadvantage is that they can be bulky. They can be in a higher bulky means, let's say for example, the manure. The manure, they can be bulky, and let's look at for example, another example is animal dung. Animal dung can also be bulky, and they can contain insect, pest, and this can cause transmission of diseases. They can harbor disease, especially the dung where it's wet. It's a very good place for insect and other disease, for example, fungi disease. Now, we move to the benefit of conservational project. Benefit of conservational project is that you keep the ecosystem, and when you're looking at the ecosystem, you keep the food chain and food web in that ecosystem intact. And this will increase diversity. Animal will reproduce, animal will be able to grow, you are going to have a larger population of different species, and also you will be able to reduce extinction, and you are able to have tourism that are coming in your country and seeing these different animal, and in that way you can generate income. We come to the end of our presentation. Please remember, you have 3 examinations. Some of you are writing in April, August, or November, but this is the guidelines that you must focus. Focus on, read through carefully in your study module, and all the best. Good luck, everybody.